I'm Kuan Zhang from Wuhan University. I'm very happy to have this opportunity to share our latest work in which we use solar-induced chlorophyll fluorescence to estimate global evapotranspiration. So here I'm the leader of the eco-hydrological processes lab, which is named as Zhang Lab, eco-hydrological processes. Okay, so this is me. I will present this work. This work has already published in Remote Sensing of Environment. The title is Solar-induced chlorophyll fluorescence sheds light on global evapotranspiration. So here is the outline. There are 4 parts. I will present one by one. First, let's look at the background. Evapotranspiration is a key hydrological component. ET links water cycle and biogeochemical processes. In global water cycle, 60% of precipitation is retained to atmosphere through evapotranspiration. So it is very important. That is also the reason why scientific community pays attention to the ET modeling. So we developed many models. There are several types of models. One of the type is the energy balance-based model. So basically we have very popular ones like the SIBO and the SEPS. Interestingly, they are all based, I think, from Europe, from European scientists. They are very good models. And the second type of model is a meteorological-based model. So they basically use either Pullman-Monti or Priestley-Taylor equation. There are also some other different equations. So this is the second type. There is also another type, the water-carbon coupling model. So basically, they model the photosynthesis, or we say the gross primary productivity. They start from evapotranspiration modeling from carbon. So that is the third evapotranspiration model. There are some problems in the prevailing ET models. As I mentioned, the first type, the energy balance-based model, it is very sensitive to land surface temperature retrieval. So basically, we need to know the surface temperature to derive the evapotranspiration. But the land surface temperature is not easy to get accurately. So that is a problem the energy balance model comes across. Well, the second type of model, the meteorological-based model, they usually need to parameterize stomatal conductance. Stomatal conductance is a measure of the stomatal aperture. So basically, water and CO2, they come through this stomatal aperture. In this kind of meteorological-based models, they need to quantify how much the stomata opens. So this is a highly complex matrix. It is kind of difficult to describe. So that is the reason why meteorological-based models, they have kind of uncertainty related to the stomatal conductance parameterization. Recently, we have this kind of solar-induced chlorophyll fluorescence measurements. Yeah, our seminar is also named after SAFE. So basically, when the canopy receives solar radiation, some of them is absorbed by the chlorophyll. So that basically goes to photosynthesis, and the solar radiation goes to carbohydrates. Some of the radiation is dissipated through the heat, and lastly, a very small proportion of radiation is emitted as this kind of fluorescence we call the solar-induced fluorescence. So this is how the SAFE is generated. After this, we know SAFE originates from the photosynthetic processes. So that is the reason why SAFE is commonly used to establish photosynthesis models. So we know SAFE can reflect photosynthesis very well, and then we assume SAFE also has great potentials to reflect the transpiration, because transpiration and photosynthesis, they share the same pathway, the stomatome. So this is where our story starts. When we did the literature review, we know some studies have already used SAFE to estimate evapotranspiration. Like in this model in 2018, they used this very simple linear equation to describe the relationship between transpiration and SAFE. So this is a very simple linear equation. There is just one parameter, alpha. The advantage of this model is it's very simple, very easy to use, just a linear regression. But the disadvantage is the relationship between transpiration with T and safe may be highly nonlinear. So this simple model may have a very big error in the estimation. There are some other studies which put this effort a little bit further. They use SIF to parameterize stomata conductance. So they use this to quantify stomata conductance. They see how big the opening of the stomata. After that, they use Monti's model to calculate transpiration. Yeah, just these two equations. The advantage of this method is they use SIF to parameterize stomatal conductance because SIF has already considered the photosynthesis processes. So it has a very tight link with the biological processes. The advantage is it can capture the stomatal coordination. So I think it should be better than some static vegetation index, like leaf area index. There is still a disadvantage in using this kind of model because the stomatal conductance remains required in such routine. As I mentioned before, stomatal conductance actually is a very complex metric. If we use this parameter, the uncertainty associated with it is inevitable. So this is the disadvantage of this model. So when we want to use SIF to estimate evapotranspiration, but the previous models, they all lack a mechanism. They just use empirical equivalence or just use SIF to parameterize stomatal conductance. They still lack a mechanism. So that is, in order to address this problem, we want to develop a SIF-based model with a physical mechanism. After development of this model, we want to use it to estimate global terrestrial evapotranspiration. So this is the aim of our study. To do this, we need to develop the model first. So evapotranspiration is basically the water returned from the Earth's surface to the atmosphere. In the terrestrial ecosystems, we use this kind of three source model. We partition evapotranspiration into three different components. The first component is the transpiration. The second one is the soil evaporation. The third one is the interception evaporation. So of these three different components, the transpiration component is tightly related to biological processes. So this is where we start the development of this model. In order to simulate transpiration using SIF, we start from the photosynthesis processes. So we use this equation GPP equals stomatal conductance to CO2 times the CO2 gradient between air and the intercellular phase. So this is just basically the Fick's law. Similarly, we can also do the same thing for transpiration. So transpiration also equals stomatal conductance to water vapor times this is the water vapor fraction deficit fraction. So this D can be quantified by vapor pressure deficit over air pressure. So we have these two equations, and then we just put them together, we just pile them up, and then we have GPP. This is basically the photosynthesis over the canopy, over transpiration, and then it equals something here, just these two different components. Let's look at this equation closely, and then we can realize that this stomatal conductance to CO2 and conductance to water vapor, they have some relationship. They have 1.6 scaling factor. So basically, we can cancel these two stomatal conductance, basically. So in this equation, GPP, it is the photosynthesis. We can link this GPP to SIF. So basically, that is how SIF is commonly used. So SIF here, we also adopt the kind of equation to describe photosynthesis. And then using these equations, what we can get is we can solve the transpiration variable here. So transpiration equals something. It is a function of safe, D, which is the VPD over air pressure. And here we have Ca minus Ci. So in this equation, we can measure safe. We can also measure this D. We can also measure Ca. So Ca basically is the air CO2 concentration. We can measure it. Here, Ci is something we don't know because we cannot measure CO2 in the leaf, the intercellular CO2 concentration. We cannot measure. So in order to estimate the transpiration using this model, we have to either replace the Ci using some known variables, or we just remove it. So here the IF safe is basically GPP. So we just use a very simple linear equation to simulate the GPP. This is commonly accepted, but there's still some uncertainty. Here, in order to simplify the model, we just use the simple linear relationship between GPP and safe. So next, we need to reduce the unknown variables from this transpiration equation. So next, let's see how we remove the Ci. Professor Farquhar from Australia, he proposed the Samatha optimization theory. So in their theory, they can quantify the CO2 gradient by using some equations here. So here, finally, they can quantify this Ca minus Ci by using Ca tau lambda C F D. So here, I will explain the variables one by one. D is what we mentioned just now. So this is the kind of vapor pressure deficit fraction. So it is related to VPD. The Ca is the air CO2 concentration. This tau is the plant compensation point. So basically, under this CO2 condition, CO2 concentration condition, the photosynthesis equals respiration. So the net CO2 uptake is zero. So this tau is related to plant type function. It can be viewed as a constant. This lambda CF is the marginal water use efficiency. It is also related to plant function type. So we can also treat it as a constant. After using this Farquhar plant optimization theory, we successfully remove this unknown variable Ci. So we can have this final equation for transpiration. Let's look at this equation here for transpiration. We have a function of safe. So basically, this is the modeling of photosynthesis. So here, this is also GPP. We can use different equations. But in our model, we just use the linear, or the very simple one, the linear equation. And here, 1.6 is a constant. Lambda CF is the marginal water use efficiency. It is also constant. D is what we can measure. We can measure VPD and derive this variable. Here, Ca is the air CO2 concentration. We can also measure that, CO2. Tau is also what we can know as long as we know the ecosystem type or the plant type function. Yeah. So here, for this transpiration model, we only have two free parameters to describe the relationship between gross primary productivity and safe. So we established this model. After this, we have a total evapotranspiration model here. So total evapotranspiration has three different components. We have already get this transpiration. The other two components, there are very mature methods to describe them. So we just adopted two mature models to simulate soil evaporation and canopy evaporation. This is the interception evaporation. We use this classic analytical gauge model. Yeah, so this is the model we developed. After that, we need to first evaluate whether they can perform well at the FluxNet size. So here we adopted this FluxNet 2015 data size. We selected 6 to 8 sites to satisfy these requirements. So here we avoid multiple towers in a remote sensing pixel because our SIF is from remote sensing. So we need to pair this pixel with the flux tower. If one pixel has multiple flux towers, we cannot determine which one can represent this pixel. So we just avoid this scenario. We also need to ensure that the data coverage of the measurements should be longer than 2 years. And the third one is we need to make sure this flux site can represent the region, to use the leaf area index to track if the tower site and the surrounding area has the similar leaf area index characteristics. So basically, that is the criteria we use to filter size. Finally, we have 68 size. These are the 68 size distribution of the globe. They represent almost all the major plant type functions. After that, we evaluated the model at the FluxNet size. We tried to push this model to the global scale. So here are all the driving variables from different sources with the contrasting spatial and temporal resolutions. Our global ET estimation is aimed at temporal resolution of 4 days. Spatial resolution is 0.05 degree. So basically, this is the temporal and spatial resolution of the safe data we adopted. So either we sample or average all the different variables to this resolution. These are the driving forces. In order to evaluate if our model can produce reliable global evapotranspiration estimate, we adopted 3 different prevailing meteorological-based ET models. The first 2 are PTGPL and GLIM. So these 2 models are basically based on Priestley-Taylor equation. And the third one is the mode 16 model. So basically, they use MODIS satellite products together with the Pengman-Montes equation. Okay, so after we have this model set up, let's see the results under the discussion here. We first evaluated the model at the FluxNet size. These are the representative size for each plant PFT. So basically, the model can perform, most of them are satisfying. If we look at the R square, most of them are around 0.7 or 8. But there are some sites like this close shrubland. The R square is only 0.1. The performance is not that satisfying for this one. And also for this woody savanna, the results is not satisfying. But considering the closed shrubland or the woody savanna, actually the vegetation, the background biomass is not that big compared with like forest, the deciduous broadleaf forest, and also the croplands. Yeah, so this model performance should also vary with the biomass because our model is basically based on SIF. SIF can well reflect the photosynthesis. And if the photosynthesis is high, like the forest, and then the model error should be low. So this is the general performance at the FluxNet size. So after that, we parameterize the model at the 68 size. And then if we want to extend this model to the global scale, we need to establish the global distribution of the 2 parameters, alpha and beta. For all the driving forces, like the meteorological forcing or the precipitation or the vegetation, we all have them. So here we need to establish the global distribution of the parameters of the model. So here is what we did. For the PFT with very limited flux size, for example, if for the closed shrubland, maybe we just have 2 or 3 sites, then we just use the average of the parameter to establish the global distribution. If we have, for a given PFT, if we have more than 5 flux size, then we use 3 different meteorological characteristics to establish the model. Here we use the multiple linear regression. we just treat the variable as the independent variable, and these three are the driving forces. So here is what we get. For this PFT, like closed shrubland, this wetland, open shrubland, we have limited flux net size. So we derive the average of these parameters. So these are the average of all the flux size of this PFT. This is the beta values. For this PFT, like cropland, we have more than 5 flux size. So we can establish the relationship between parameter and these different driving variables. These equations are applied to global scale to establish the parameter of alpha and beta. For this PFT, because we have limited flux net size, so we just assign constant value to all the closed shrubland over the globe. There is also one thing to note. Here, beta is the intercept to describe GPP and safe. Because we know when safe is 0, GPP should also be 0. So to avoid some negative photosynthesis rate, here, if beta is negative, we force it to be 0. Yeah, so this is one note. After this treatment, we establish the global distribution of the parameters. So this figure A and B represent the distribution of parameter alpha and beta. The top one is alpha. We compared this alpha with some reported values. Most of them are acceptable. But there's still some very high values, like in the northern hemisphere, somewhere here in the boreal ecosystem. The alpha is very high. In this area, they can do a little higher than the normal values. Yeah, so maybe there's still some uncertainty in our model to establish this global distribution. Regarding this beta, it is very similar to what is reported before. Yeah, so this is the global distribution of the parameters. After we get these parameters, we can just push the whole model to the global scale because we have all the global meteorological forcings, precipitation, and also vegetation. So we just evaluate the global scale evapotranspiration. This map presents the spatial distribution of global mean annual evapotranspiration. It is close to our knowledge in the tropical region like this Central Africa and also Indonesia, this is Southeast Asia, and also the Brazil, the Amazon forest. We generally have very high values over the tropical regions. In the high latitude, like this North America, and also here, we have very low values. So it is close to our common knowledge. We also draw a figure of the PDF of this global mean evapotranspiration. So we produce a safe-based evapotranspiration, which is close to our knowledge. We also evaluate the trend of this global annual evapotranspiration because our safe has a short time coverage. So we evaluated the global evapotranspiration from 2003 to 2018. During this period, the whole global evapotranspiration showed a slightly increasing trend, though this trend is not significant. We also evaluate the global mean evapotranspiration over this period. It is about 625 millimeter per year. Next, we compared our product with other ET evapotranspiration products. This is the difference between our ET estimation and the GLEAM product. If we look at the map, we can see some pronounced difference over the tropical region, like this Central Africa and also Southeast China. And also here, the difference can be as high as like 6 watts per square meter. The difference is very high. And also here in the United States, the difference is also very high. If we look at the PDF, So the difference is mostly centered around 0. So for these two products, our ET and the Gallium ET, they are kind of similar, even though there are some difference here. We also compared our data with the PTGPL model. So here we can still see some very high difference. If you look at the PDF, it has two peaks. So basically these two products should have some major difference. We also evaluated the difference between ours and the mode 16 ET. The mode 16 evapotranspiration product has contributed a lot of knowledge to hydrological research regarding evapotranspiration. But their product only based on vegetation area. So for these, like Sahara Desert and also some other desert, they did not evaluate. The difference between ours and the mode 16 is very huge. We can see the difference like almost the whole Africa. We see much difference, and also other regions of the tropical area. Regarding the PDF, the difference is also very high. It also has two peaks. Yeah, the difference is pronounced very, very clear. It's very large. Okay, let's conclude here. Our work has contributed in two aspects. The first is we produce the global ET dataset, mainly driven by SAFE. To the best of our knowledge, I think this is the first evapotranspiration using SAFE. And the second is we evaluate the global evapotranspiration trend over the period of 2003 to 2018. The global mean ET value is 625 millimeter per year from our research, and there is also a slight rising trend in ET during this period. After that, we also want to have a small summary here. The advantage of our model is very simple in structure, but with the physical mechanism. In our derivation of the transpiration, each step has a strong physical basis. So our model is, we can say, it is physically based. It has a physical mechanism. The second one is we avoid parameterizing stomatoconductance. We cancel that out. So that can help us reduce the possible uncertainty related to this kind of stomatoconductance and parameterization. We still have some limitation. We use a very simple equation between GBP and SAFE. That may bring some error. Yeah, so that may be something we can further improve. The second one is though we estimated the ET using SAFE, we believe SAFE has a very good quality to represent plant biological processes. But our estimation, our global transpiration estimation, are a little high. That is what we need to improve in the future, possibly. Okay, that's it. Thank you for your attention. Thank you, Julian, for the warm invitation.